Hello again. Chapter 5, we are going to talk about microbial metabolism. I thought you might enjoy this picture of Edward puppy hands. What do you think these puppies are doing right here? Metabolism is the sum of chemical reactions in an organism. Ultimate function of metabolism is to reproduce the organism, and in order to do that, we have to make energy. We have two processes by which we do metabolism. The first, catabolism, provides energy and the building blocks for anabolism. Catabolism breaks down larger molecules into smaller ones. In doing so, it releases energy, and so catabolic reactions are considered exergonic, meaning energy exits. The other metabolic reaction is anabolism. This uses energy and building blocks to build large molecules from smaller ones. Anabolistic reactions are endergonic, meaning they require more energy than they release. The role of ATP in catabolic and anabolic reactions is shown here. Catabolic reactions transfer energy from complex molecules to form ATP. Catabolic reactions then break down those molecules into the simple molecules such as glucose, amino acids, glycerol, and fatty acids, making ATP in the process. That ATP and the simple molecules then undergo anabolic reactions and transfer the energy from ATP to build larger, more complex molecules, such as starch, proteins, and lipids. In doing so, we use an ATP and will release more heat than is produced. And you can see that this is a cyclic relationship between catabolism and anabolism. Metabolic processes are guided by 8 elementary statements. First, every cell acquires nutrients somehow, some way. Metabolism requires energy from either light or from catabolism of nutrients. Energy is stored in the form of adenosine triphosphate, ATP, the only form of energy a cell can use. Cells catabolize nutrients to form precursor metabolites. These precursor metabolites, the energy from ATP and enzymes are used in anabolic reactions to build what the cell needs. Enzymes plus ATP then form macromolecules. The cells grow by assembling these macromolecules, and cells reproduce once they have pretty much doubled in size. A metabolic pathway is a sequence of enzymatically catalyzed chemical reactions within a cell. Metabolic pathways are determined by what enzymes are present within the cell. Enzymes are encoded for by the genes and structurally originate from proteins. This means that all enzymes are structurally proteins or came from proteins. That does not mean all proteins act as enzymes. In chemical reactions, the collision theory states that for the chemical reaction to occur, atoms, ions, or molecules must collide with each other, and they need enough energy in that collision to disrupt the electronic configurations. This is known as the activation energy of the reaction. The reaction rate is the frequency of collisions with enough activation energy to bring about a reaction. Reaction rates can be increased by the presence of enzymes or by increasing the temperature or pressure in which the reaction occurs. Here's your picture of the collision theory. The orange and green balls smack into each other, and we have a change in each of those balls. And in order to do that change, we have to have that specific amount of energy in order to cause the reaction to occur. The balls may gently touch, but there would not be enough energy to actually cause the chemical reaction to occur. Here it's shown in graphic form. If AB without an enzyme goes to product A plus B, it would take this much energy to complete that reaction. However, if we introduce an enzyme into the reaction, it will only take this much energy in order to get A plus B. Therefore, enzymes are globular proteins that promote chemical reactions by lowering the activation needed for the reaction, but are not consumed themselves in the reaction. They come out on the other end of the reaction unchanged. Therefore, many times we will write the name of the enzyme above our reaction arrow because they come out unchanged at the end. So reactant A plus reactant B plus the enzyme will go to product AB plus the enzyme exactly as it was before. Enzymes are biological catalysts. They are specific for a chemical reaction but are not used up in that reaction. Most of our enzymes actually exist in what we call the apoenzyme form. This is the actual protein component of the enzyme. Most of the time we will need some sort of cofactor or coenzyme that will combine with the apoenzyme to create a holoenzyme, which is the active enzyme molecule that will cause a chemical reaction to occur. In table 5.2 on page 129, you have some of these coenzymes and cofactors. Some of the ones we will talk about will be nicotinamide adenine dinucleotide NAD+, nicotinamide adenine dinucleotide phosphate NAD+, flavin adenine dinucleotide FAD, and coenzyme A. In this figure, the apoenzyme is unable to bind with the substrate. The substrate does not match the shape of the protein. But when we add the coenzyme to the apoenzyme, you'll see that the substrate now can fit nicely, just like a piece of a puzzle, into the holoenzyme. And this will then cause the chemical reaction to occur. Enzymatic activity varies with the enzyme, but the turnover number, meaning how many substrates can an enzyme act on in a given time, is generally 1 to 10,000 molecules per second. So, for example, the enzyme fructose 1,6-biphosphate atolase will take fructose 1,6-biphosphate, form the enzyme substrate complex, and create glyceraldehyde 3-phosphate, known as G3P, and dihydroxyacetone phosphate. It can do this as long as there is substrate present and the enzyme is active. Here's a picture of a substrate binding to its holoenzyme, forming the enzyme substrate complex. This strains bonds within the substrate to either create or break bonds, changing the substrate in a chemical reaction. So the mechanism of enzymatic reaction is this. The surface of the substrate contacts the active site of the holoenzyme. An enzyme substrate complex is formed. This is a temporary intermediate compound. The substrate molecule is strained in such a way that it is transformed by the rearrangement of the existing atoms within the bonds, and the transformed substrate molecules, the products, are released from the enzyme's active site because it no longer fits the active site. It has changed shape. The unchanged enzyme is now free to interact with another substrate molecule. An analogy of this would be the junior high dance. You go to the junior high dance, and what do you see? Usually boys on one side, girls on the other. You want dancing partners. Here I come as the chaperone, and I take these two able but unwilling molecules, David and Joan, and introduce them to each other and form a dancing couple. I can continue to do that until I've matched up all the willing and unable molecules to be willing and able molecules. However, do you want me to do this job on Monday morning in the hallway of the junior high? Or would you like some way to inactivate me? On table 5.1 on page 128, here are some of the major classes of enzymes. Anytime you see ASE at the end of a word, it means enzyme. Also, zyme, Z-Y-M-E, will also indicate that the substance is an enzyme. The front part of the word is either what type of reaction occurs or the substrate. For example, hydrolase causes hydrolysis, catabolic reactions, and a good example of this would be a lipase. The lipase, lip means the substrate is a lipid. Isomerases rearrange the atoms within a molecule. This is neither catabolic or anabolic, but simply rearranges it, forming an isomer. So we do not really change any atoms within the molecule, just change how they're arranged. Ligases or polymerases join molecules together, and these are anabolic reactions. A lyase will split a chemical into smaller parts without using water. The oxyreductase transfers electrons or hydrogen atoms from one molecule to another in redox reactions. And transferase does just what it says, transfers one molecule or atom to another. Factors that can influence enzymatic activity include temperature, pH, substrate concentration, the presence of any inhibitors. Temperature and pH both denature proteins. What I mean by denaturing is it literally tears the protein structure apart. I do this every time I fry an egg. The albumin in the egg white, when I put it on the hot skillet, goes from a liquid to a solid state. This is coagulation. How this occurs is the hot temperature actually denatures the albumin of the egg white. If I took a raw egg and put it into hydrochloric acid with a pH of 1, it would do the same thing. A basic pH would basically do the same thing as an acidic pH. Most enzymes will have an optimum temperature at which they operate at the best level. For example, this enzyme shows that its best level is actually at about 37 degrees Celsius, which is body temperature of the human. You'll notice that as heat increases, the activity of the enzyme drops off significantly rapidly. However, colder temperatures, while the enzymatic activity decreases, it actually takes a longer time to completely inactivate the enzyme to zero activity. So colder temperatures, while they do affect enzymatic activity, take a longer time to completely denature the proteins. pH, most enzymes will have an optimum pH range at which they like to operate. For example, this one has a maximum activity at about a pH of 5. What would our enzyme's maximum pH level be? How much substrate you have also affects the activity of the enzyme. At a certain point, increasing the amount of substrate will not increase the rate of reaction. This is because all the enzyme active sites are filled with substrate, and adding more simply isn't going to make the enzymes work any faster. They are working at their maximum capacity with the substrate. And this point where the rate of reaction no longer increases is known as the point of saturation. Doesn't matter how much more substrate you put in, we are at the peak of efficiency. I also said that if inhibitors are present, this can affect the rate of reaction of enzymes. If the enzyme is an active enzyme, a holoenzyme, the substrate binds, we get the chemical reaction, we get the product. However, if an inhibitor is present, it can stop the binding of the substrate to the enzyme. And we do this in two ways. This shows competitive inhibition. In this particular case, this yellow substance binds to part of the active site on the enzyme and therefore blocks the ability of the substrate to bind. It is competing for the active site with the normal substrate. Increasing substrate level, however, may reverse the inhibitor. So if my substrate is this bluish thing and the competitive inhibitor is the red box, I can actually knock off the competitive inhibitor by simply putting in more substrate. The enzyme says, I've got more of this stuff over here, the substrate, to work with than I do the inhibitor. I'll throw the inhibitor off and start working with the substrate. Non-competitive inhibition, however, is similar, but we do not bind to the active site. We have an allosteric site on the holoenzyme. When the non-competitive inhibitor binds to this, it alters the active site in such a way that the substrate can no longer bind to its enzyme. This works with negative feedback many times. So here we have a cascade, an enzymatic pathway where enzyme 1 binds to substrate 1, produces an intermediate product A, that binds to enzyme 2, which produces an intermediate product B, which binds to the third enzyme, which we wind up with the end product. The end product goes back to enzyme 1, where it binds to the allosteric site and shuts the pathway down. As long as I have plenty of my end product, I don't need to continue to make it. So this is negative feedback loop by non-competitive inhibition. Basically, the product shuts the first enzyme down, shutting down the enzymatic cascade. Oxidation-reduction reactions. Oxidation refers to the removal of electrons from a substance, while reduction is the gain of electrons. The way to remember this is Leo goes Ger. Losing electrons is oxidation, Leo. Gaining electrons is reduction, Ger. Usually an oxidation and reduction reaction occur at the same time. Somebody is going to lose the electron and somebody needs to pick it up. An redox reaction is when an oxidation reaction is paired with a reduction reaction. For example, here we have an electron donor. It gives up the electron to the electron acceptor. The donor, therefore, is oxidized because it lost the electrons, and the acceptor is reduced because it gained the electrons. Oxidation-reduction reactions, therefore, transfer electrons from an electron donor to an electron acceptor. These reactions almost always occur simultaneously. Cells use electron carriers to carry electrons, most often in the form of hydrogen atoms. Remember, hydrogens only have normally one electron. So, we can simply give up the hydrogen atom and we give up the single electron. There are three important electron carriers that we'll discuss in cellular metabolism: nicotinamide adenine dinucleotide, NAD, nicotinamide adenine dinucleotide phosphate, NADPH, and flavin adenine dinucleotide, FAD, which becomes FADH2. NAD and NADP both can accept a single electron. FAD can accept two. In biological systems, since the electrons are most associated with hydrogen atoms, these are often called dehydrogenations. We lose the hydrogen. For example, I have an organic molecule that includes two hydrogen atoms. These two hydrogen atoms are given up to NAD as the electron carrier. My organic substance, therefore, is oxidized because it lost the electrons in the form of hydrogens, and NAD gained a hydrogen atom, hence NADH, but one of the hydrogens will actually give up its electron to NADH and go off as the hydrogen ion. NADH is now a reduced electron carrier because it received the electrons. In aerobic respiration, glucose will be oxidized and lose hydrogen atoms to oxygen molecules eventually. Therefore, oxygen is reduced by gaining the hydrogens from the glucose, and this oxygen then will form water. Glucose loses the hydrogen atoms, eventually forming CO2, while oxygen gains the hydrogen, forming water. In this process, we also make ATP. Organisms release energy from nutrients. This energy then can be concentrated and stored in high-energy phosphate bonds within ATP. Phosphorylation is when an organic phosphate is added to the substrate, and cells will phosphorylate ADP, adenosine diphosphate, to ATP in three ways: substrate-level phosphorylation, oxidative phosphorylation, and photophosphorylation. We'll talk about each one of these in turn. Anabolic pathways use energy of ATP by breaking a phosphate bond on ATP, converting it back to ADP. So therefore, ATP is a renewable source of energy, as long as we can actually capture energy from cellular respiration, from the breakdown of nutrients, put the phosphate back on ADP, and create out ATP again. This is an example of substrate-level phosphorylation. Here we have the holoenzyme and its substrate, phosphoenol pyruvate, or PEP. I like the acronym. Magnesium is needed here as a cofactor on the holoenzyme, and it binds to PEP, takes a phosphate off of the phosphoenol pyruvate, adds it to ADP, makes ATP, and generates out what's left as pyruvic acid. This is the last step of glycolysis. Oxidative phosphorylation, energy will be released from the transfer of electrons, oxidation, from one compound to another that is reduced, and is used to generate ATP within some form of an electron transport chain. This is a picture of our electron transport chain that we have in our cells. We will go over it in more detail in just a moment. But basically what will happen is electron carriers such as NADH bring in hydrogen and electrons to this series of enzymatic reactions and eventually generates out ATP by using an enzyme called ATP synthase. Photophosphorylation, as you might expect, is energy from light. Light causes chlorophyll to give up electrons to an electron transport chain. Energy released from the transfer of these electrons then of the chlorophyll through a system of carrier molecules is used to generate ATP using ATP synthase, just like we saw. But in this case, it's not an electron carrier that brings in the energy, it's light. Here's a simplified version of photosynthesis. Again, we'll talk about this more in detail later on. Carbohydrate catabolism. The breakdown of carbohydrates within cells to release energy to generate ATP is broken down into three stages: glycolysis, the citric acid cycle, which the old term was the Krebs cycle, and the electron transport chain. We'll look at each of these in detail. Many organisms oxidize carbohydrates as a primary energy source for these anabolic reactions to make ATP. Glucose is the most common carbohydrate used, and so we will look at glucose catabolism. Glucose can be catabolized by two processes: cellular respiration and fermentation. Here is cellular respiration and fermentation in a simplified flowchart. Again, we'll look at each of these steps in detail. Let's start with glycolysis. Glycolysis occurs in the cytoplasm of most cells. It will involve splitting a 6-carbon glucose into two 3-carbon sugar molecules, pyruvic acids. It involves substrate-level phosphorylation, the direct transfer of phosphate between two substrates, and you get a net gain of 2 ATP molecules, 2 molecules of NADH, 2 precursor metabolite pyruvic acids, and 2 waters. Here is the preparatory stages of glycolysis. Glucose will be taken by an enzyme, hexokinase, to glucose 6-phosphate. Where did we get this phosphate? Oh, you'll notice something here. Wait a minute, I just used an ATP. I thought the whole process was that we were going to make ATP. Well, it's kind of like when you make money. In order to make money, what do you have to have as an initial investment? You've got to have some money. So we're going to have to use some energy to begin with. That's okay. We'll get it back. Glucose 6-phosphate then in turn is metabolized to fructose 6-phosphate, which eventually goes to fructose 1,6-diphosphate. And you'll notice we use another ATP. We are now down 2 ATP. Keep that in mind. This is the energy investment and lysis stage because fructose 1,6-diphosphate in step 4 will be split into 2 3-carbon products, glyceraldehyde 3-phosphate, G3P, and dihydroxyacetone phosphate. You'll notice in step 5 that there is actually an enzyme here, an isomerase, which converts the isomer DHAP to its mirror image of glyceraldehyde 3-phosphate. Therefore, for each glucose that enters glycolysis, you wind up with 2 glyceraldehyde 3-phosphates at the end of this investment stages. We then will take the glyceraldehyde 3-phosphate and go through another series of enzymatic reactions to finish glycolysis in the energy conserving stage. Here, you'll notice a few things. 1, 2 glyceraldehyde 3-phosphates, which that should say glyceraldehyde, not glucose, is oxidized to 2 pyruvic acid molecules. You get a total of 4 ATP for every original glucose in step 7 and step 10. You get 2 NADHs produced out of step 6, and you have 2 water molecules produced out of step 9 as a byproduct. This picture actually recognizes all of the enzymes in glycolysis. You do not have to memorize these. I simply put this slide in to remind us that each one of these steps in glycolysis has an associated enzyme. What you really need to understand about glycolysis is this. We start with glucose plus 2 ATP as our initial investment, plus 2 ADP, plus 2 phosphate groups, plus 2 NADH+. This will go to 2 pyruvic acids, 4 ATP, plus 2 NADH, plus 2 hydrogen ions that will form 2 water molecules. Glycolysis occurs in the cytosol of the cell. They are anaerobic reactions. Does not matter if oxygen is present or not. Glycolysis will run as long as there is substrate and enzymes present. A net gain of 2 ATP molecules. Now you may say, wait, we had 4 ATP. Yes, but remember, we used 2 to begin with. We have to pay those back. So we gain 2 ATP overall. 2 pyruvic acid molecules are produced, 2 NADH molecules, and 2 water molecules are produced. We do have some alternative pathways to glycolysis if the cell has the enzymes that can run these pathways. On page 143, you have a figure of the pentose phosphate pathway. Instead of a 6-carbon sugar, the pentose phosphate pathway uses a 5-carbon sugar and NADPH as the carrier. It does use many of the same enzymes as glycolysis, but simply starts with the 5-carbon sugar. The Internodordoff pathway, shown on page 144, this produces NADPH and ATP, but it does not involve any of the enzymes of glycolysis. This is a completely different pathway than pentose phosphate and glycolysis. Some examples of bacteria that use the Internodordoff pathway are Pseudomonas, Rhizopum, and Agrobacterium. The Rhizopum and Agrobacterium are important in plant agriculture. The pentose phosphate pathway is primarily an anabolic pathway that utilizes the 6 carbons of glucose to generate 5 carbon sugars that enter the pathway and what we call reducing equivalents. Functions to generate the reducing equivalents in the form of NADPH is for reductive biosynthesis reactions within the cell. What this does is provide the cell primarily with ribose 5-phosphate. This is important in our cells for the synthesis of the nucleotides and nucleic acids. So remember, ribonucleic acid, deoxyribonucleic acid. Both started with ribose, deoxyribose. Although not a significant function, the pentose phosphate pathway can operate to metabolize dietary pentose sugars that may be delivered to the cells, as well as digestion of nucleic acids by rearranging the carbon skeletons of these dietary carbohydrates into glycolytic and gluconeogenic intermediates. Here's a simplified version of the pentose phosphate pathway. And again, note here, NADPH and the ribose 5-phosphate that will be used for synthesis of nucleotides in the nucleic acids. The internodordoff pathway, an alternate pathway that catabolizes glucose to pyruvate using a set of enzymes completely different from those used in either glycolysis or pentose phosphate pathway. The internodordoff pathway, when it does occur, only occurs in prokaryotic cells. It only yields one ATP, one NADH, and one NADPH for each glucose. And all of the Archaea kingdom use the internodordoff pathway exclusively. This is the internodordoff pathway. Under no circumstances, memorize this pathway. Now, we have pyruvic acid from glycolysis. Is there still energy present within the bonds of pyruvic acid? And the answer is yes. So, oxidation of molecules liberates electrons for electron transport chain. But we're going to get some more energy before we get there. ATP will then be generated by oxidative phosphorylation in the electron transport chain. So how do we get there? For each original molecule of glucose that is utilized in carbohydrate catabolism, the synthesis of acetyl-CoA results in 2 molecules of acetyl-CoA, 2 molecules of CO2, and 2 molecules of NADH. This is what's known as the intermediate step between glycolysis and the citric acid cycle. Pyruvic acid from glycolysis is oxidized and decarboxylated. We take off a carbon, forming the CO2. Coenzyme A attaches to the 2 carbons left to produce acetyl-CoA. Because pyruvic acid is oxidized, somebody's got to pick up the electron. Here's our friend NADH+. So for each pyruvic acid, we get an NADH, CO2, acetyl-CoA. How many pyruvic acids did we get from glycolysis? 2. This figure on 136 shows what happens with each of the atoms as we move through the intermediate step. Now, we have the acetyl-CoA. Is there still energy in the bonds? Yes. Now we will enter the citric acid cycle, or Krebs cycle. I will refer to it as the citric acid cycle. Here we're going to oxidize the acetyl-CoA to produce NADH, FADH2, and some ATP. There's still a great amount of energy that remains in the acetyl-CoA bonds, and the citric acid cycle will transfer much of this energy to the coenzymes NAD and FAD. This occurs in the cytosol of all prokaryotic cells, but within the matrix of the mitochondria in eukaryotic cells. So there is a difference here of where they occur. 6 types of reactions occur in the citric acid cycle: anabolism of citric acid, isomerization reactions, hydration reactions, redox reactions, decarboxylations, and substrate level phosphorylation. If we go back, you'll notice acetyl-CoA enters the citric acid cycle and combines with a molecule known as oxaloacetic acid. This forms citric acid, hence the citric acid cycle. Citric acid cycle then is converted to isocitric acid, which goes to alpha-ketogluteric acid, which continues around the ring. You'll notice in step 3, we are going to get an NADH and a CO2 produced. In step 4, we get another NADH and a CO2 produced. When succinyl-CoA goes to succinic acid, we get an ATP produced. When succinic acid goes to fumaric acid, FAD becomes FADH2. And finally, when malic acid returns to oxaloacetic acid, hence the citric acid cycle, we get a third NADH. So for each acetyl-CoA that enters, we get 3 NADHs, 2 CO2s, 1 FADH2, and 1 ATP. So for each original molecule of glucose, because remember you get 2 pyruvic acids, therefore 2 acetyl-CoAs, for each original molecule of glucose that is utilized in carbohydrate catabolism, the citric acid cycle results in 2 molecules of ATP from the original glucose, 2 molecules of FADH2, 6 NADHs, and 4 molecules of CO2 as a waste product. Now, we have all the NADHs and FADH2s. Is there energy in those? Yes, because they are the electron carriers. And so they will now enter the electron transport chain. This is where we have the most significant production of ATP. It is a series of redox reactions known as the electron transport chain, with a total of anywhere from 28 to 34 ATP molecules formed from the one original molecule of glucose that started this journey in glycolysis. The electron transport chain is a series of carrier molecules that are in turn oxidized and then reduced as electrons pass down the chain from one to another, finally going to a final electron acceptor. Energy that is released during the electron's movement can be used to produce ATP by the process chemoosmosis that pumps protons, specifically hydrogen ions, across a membrane, establishing a hydrogen ion protein gradient and using ATP synthase enzyme and the hydrogen gradient to generate energy to put phosphate on ADP and make ATP. The electron transport chain occurs across the cristae of the mitochondria, that inner membrane of the mitochondria in eukaryotic cells, and across the cytoplasmic membrane of prokaryotes. Prokaryotes don't have a mitochondria. Electron transport occurs across the membrane. The only membrane we have is the cell membrane in prokaryotic cells. There are 4 classes of carrier molecules in electron transport chains. The flavoproteins that are derived from vitamin B12 or riboflavin, and this includes flavin mononucleotide or FMN. The ubiquinones, also known as coenzyme Q, and the cytochromes, that is actually derived from the iron-containing heme group. There are several of these designated by a lowercase letter or in combination with a number. They are in the chain not in alphabetical order, but they were given their designation in the order they were discovered. You do not need to memorize in what order the cytochromes fall. Some electron transport chains also have metal-containing proteins. For example, in this one, the second carrier molecule is iron sulfide. This one shows our electron transport chain within our mitochondria. NADH will give up the hydrogen and electrons to FMN, which will be reduced. FMN then oxidizes, releasing the electrons to coenzyme Q, and then flows down through the cytochromes to finally being taken on by oxygen as the final electron acceptor. Here it is across the membrane. FMN receives the NADH and a hydrogen ion. It pumps the 2 hydrogen ions across the membrane and gives the electrons to coenzyme Q. FADH2, if it can enter the chain, will give up its electrons to coenzyme Q and its hydrogens to cytochrome B. This will pump the hydrogens across to the other side of the membrane. From the NADH, we have 2 hydrogens here, 2 hydrogens here, and 2 hydrogens here coming across the membrane. From each FADH2 molecules, we would have 2 hydrogens here and 2 more hydrogens here. Each NADH, therefore, will produce 3 ATP because a pair of hydrogen ions will run through ATP synthase and take phosphate, put it on ADP to generate out ATP. As the hydrogen pairs flow through, they will combine with oxygen and form water. For each FADH2 that enters the chain, we have 2 pairs of hydrogen ions that can go through ATP synthase. Therefore, for each FADH2, we should get 2 ATP. This does not always happen. Therefore, you may have a range from anywhere from a total of 24 to 34 ATP generated along the electron transport chain. This is the highly simplified version. We create a hydrogen ion concentration gradient across the membrane, be it the cell membrane of prokaryotic cells or the inner membrane of the mitochondria. The hydrogen flow through ATP synthase in pairs. For each pair, we get an ATP. So, if the final electron acceptor of the electron transport chain is molecular oxygen that then forms water, we undergo aerobic respiration. It requires oxygen. Anaerobic respiration, however, the final electron acceptor in the electron transport chain is not oxygen. It is something else. The yields of ATP are therefore less than aerobic respiration generally because only part of the citric acid cycle will operate under anaerobic conditions. There are certain steps of the CAC that need oxygen. Some of the anaerobic electron acceptors would be things like NO3 or nitrate that would go to nitrite, nitrogen gas, and water. The sulfate ion, which would go to hydrogen sulfide and water, and the bicarbonate ion, which goes to CH4 or methane gas and water. This table shows where each step of carbohydrate catabolism occurs in both the eukaryotic and prokaryotic cell. We've mentioned each of these as we went through. This table on page 142 shows how many ATP are produced in each of the stages of carbohydrate metabolism, how many are used, how many NADH and FADH2s are produced. So, 4 ATP are produced in glycolysis, but remember, used to, there are 2 ATP produced in the synthesis of acetyl-CoA and the citric acid cycle. Electron transport chain, if all the NADH and FADH2s go through, we would get 34 for a total of 40, but again, subtracting the 2 we used way back there in glycolysis, we get a net gain of 38 ATP from 1 glucose. If anybody knows where I can put in $1 and get $38 back in an investment, please see me in class. This table shows where ATP are produced by substrate level phosphorylation and oxidative phosphorylation. You do not need to memorize this chart, but be aware that glycolysis and citric acid cycle are substrate level, while the electron transport chain is oxidative phosphorylation. The other way that we can utilize glucose is through fermentation, and there are several definitions depending upon your use of the sugar. One definition is any spoilage of food by microorganisms is considered fermentation. Another general use is any process that produces alcoholic beverages or acidic dairy products is also fermentation. A common definition used in industry is any large-scale microbial process that occurs with or without the presence of air. Fermentation is anaerobic, but the reactions of fermentation can occur in the presence of oxygen. It just doesn't need oxygen. The scientific definition of fermentation is that fermentation releases energy from the oxidation of organic molecules. It does not require oxygen. It does not use the citric acid cycle or electron transport enzymes in any way. It uses an organic molecule, either an alcohol or an acid, not oxygen, as the final electron acceptor. Sometimes cells cannot completely oxidize glucose by aerobic cellular respiration alone. Cells also require a constant source of NAD+. These molecules cannot be obtained simply by using glycolysis and the citric acid cycle. Fermentation pathways then provide cells with alternative sources of NADH+. So, partial oxidation of sugar or other metabolites to release energy using an organic molecule from within the cell as the final electron acceptor. Here's the pathway of fermentation. This occurs in our body. If pyruvic acid at the end of glycolysis does not have oxygen to enter the citric acid cycle, we then will convert pyruvic acid to lactic acid. Anyone a marathon runner? Why do marathon runners hit the wall? The reason why is the lactic acid builds up so much in their muscle cells that they finally become so acidic the muscle cells quit working. We also can produce acetaldehyde, which eventually goes to ethanol or alcohol. So the lactic acid and ethanol would be our final electron acceptors. For example, Streptococcus, Lactobacillus, and Bacillus all produce lactic acid as the end product of fermentation. Saccharomyces, which is the common bread yeast, produces ethanol and CO2. Why does bread dough rise? These other bacteria, which are some examples, produce a variety of acids and or alcohols. For example, you have some that produce ethanol, lactic acid, succinic acid, acetic acid. Some produce formic acid, acetone, isopropyl alcohol, acetic acid, and in fact, Propionibacterium is one of the bacteria that produce acetic acid or vinegar. If the fermentation pathway in the cell produces only alcohol, such as ethanol and CO2, it is an alcohol fermenter. If it produces only lactic acid, such as the Lactobacillus bacterium, it's a homolactic fermenter. If it produces lactic acid and others, it's a heterolactic fermenter. Here's some industrial uses for different types of fermentation. So, if we want ethanol as our end product, we start with Saccharomyces, which is a yeast. And we can use different starting material. If we want to make wine, we're going to start with grape or other fruit juices. If we want beer, we start with malt extract. And if we want ethanol fuel, we can start with a variety of agricultural waste products, such as corn or grasses. Acetobacter is another one of those bacteria that produce acetic acid or vinegar. The starting material, however, is ethanol. Hopefully you'll remember back to chapter 1 with Pasteur, trying to figure out why the wine, the ethanol, spoiled to vinegar. He realized that the Saccharomyces yeast made the wine, but the Acetobacter that was also in there took the ethanol produced by the yeast and created vinegar. Many of the lactic acid producers produce a number of our food products for us, such as cheese, yogurt, sour cream, rye bread, sauerkraut, and even summer sausage. If you really want to think about it, next time you eat some summer sausage, realize it's nothing more than fermented meat. In the laboratory, we will use the carbohydrate fermentation test. This test media has a pH indicator, and if it goes from red to yellow, it means that acids were produced. Therefore, fermentation occurred of that particular sugar. The small Durham tube, again, is in there to see if we produce any type of gas. This could be CO2, hydrogen gas, etc. You'll find a small bubble in the Durham tube if gas is produced during fermentation. So here's the overview of both cellular respiration and fermentation. The key concept here is to produce energy from glucose. Microbes can use two general processes, respiration and fermentation. Both usually start with glycolysis but follow subsequent different pathways. Some cells can do both respiration and fermentation, some only fermentation, some only cellular respiration. Again, it depends upon what enzymes the cell produces. Here's a comparison of aerobic and anaerobic respiration and fermentation. One thing to note is that aerobic respiration is the highest producer of ATP. 38 in prokaryotes, anywhere from 28 to 36 or so in eukaryotic cells. Anaerobic respiration, depending on the final electron acceptor, you'll have anywhere from 2 to 36 ATP produced. However, fermentation only produces 2, so it is the least efficient ATP production pathway. Can lipids be used for energy production? They can, but they take a lot of initial investment of energy on the front end in digestion. We can break the lipids down into the 3-carbon sugar glycerol and the 3 fatty acid chains. The glycerol, through enzymes, can be converted to glyceraldehyde 3-phosphate. Do the energy-conserving stages of glycolysis care where the glyceraldehyde 3-phosphate came from? No, but now instead of a total of 4 ATP, you only get 2 ATP produced. The fatty acids, after they've undergone a process known as beta oxidation, can be converted to acetyl-CoA that can enter then into the citric acid cycle. Here's the breakdown of the lipids through lipase to glycerol converted to G3P, and the fatty acid chains undergoing beta oxidation and making acetyl-CoA. Proteins can be used as an energy source, but most cells do not like to do this. Primarily, cells like to use proteins as building blocks, not as an energy source. Part of the reason for this is proteins have to undergo a large amount of processing before they can enter the different stages of respiration. Some amino acids can be converted to pyruvic acid, some to acetyl-CoA, some to the intermediate products found in the citric acid cycle. One of the things that does have to occur, however, for us to use a protein or an amino acid as an energy source is deamination. We have to take off that NH2 group, the amino group. This then can convert the rest of the product left to acetyl-CoA. Photosynthesis, using light energy to create ATP. I showed you this picture prior of the chromatophores, of the invaginations of the cell membrane of this particular purple bacteria. This is where the thylakoids for photosynthesis will be located within these photosynthetic bacteria. Many organisms synthesize their own organic molecules from inorganic carbon dioxide. Most of these organisms capture light energy and use it to synthesize carbohydrates from the CO2 in the environment and water by a process called photosynthesis. Chlorophylls are important to organisms because they capture the light energy with these pigmented molecules. Chlorophylls are composed of a hydrocarbon tail attached to a light-absorbing active site centered around a magnesium ion. The active sites of chlorophyll is actually structurally similar to the cytochrome molecules in the electron transport chain. Structural differences within the chlorophylls cause absorption of different wavelengths of light. We think primarily of the yellow-green spectrum with our plants, but that purple bacterium I showed you actually absorbs light waves in the blue-violet spectrum. In eukaryotic cells, the chlorophyll is contained within the thylakoids located within the chloroplasts. The thylakoid membrane has an electron transport type chain and ATP synthase. The arrangement of the molecules of chlorophyll and other pigments form light harvesting matrices in the thylakoids. They are embedded in the membranes of the thylakoids, and the thylakoids then are arranged in stacks within the chloroplast called grana. The stroma is the space between the outer membrane of the grana and the thylakoid membranes. In prokaryotic cells, the thylakoids are found in the invaginations of the cytoplasmic membrane, and in eukaryotic cells, the thylakoids form the inner membrane of the chloroplasts. Here's one of these thylakoids with the embedded chlorophyll. This is a molecule of chlorophyll. Again, a long chain of carbons and hydrogens attached to a head surrounding a magnesium ion. There are two types of photosystems that we see in photosynthesis, photosystem 1 and photosystem 2. Photosystems will absorb light energy and use redox reactions to store energy in the form of ATP and NADPH. There are light-dependent reactions that depend on harvesting the light energy, and then there are light-independent reactions that synthesize glucose from carbon dioxide and water. The light-dependent reactions occur as electrons move down the chain. Their energy is used to pump protons across the membrane. That should sound familiar. Photophosphorylation then uses the proton motive force, the movement of hydrogen ions from a high area of concentration to a low area of concentration through ATP synthase to generate ATP. Photophosphorylation can be either cyclic or non-cyclic, depending upon who is the final electron acceptor. Light strikes the chlorophyll and is bounced to the reaction center chlorophyll in the middle of the thylakoid. This excites chlorophyll, and the chlorophyll then will give up electrons to the electron transport chain. Here we can see photosystem 1 and photosystem 2. We'll talk about each of these separately. In photosystem 1, the light-dependent reaction is cyclic. When the light strikes the chlorophyll in the photosystem 1 thylakoid, we go through a series of electron carriers back to chlorophyll. Chlorophyll actually becomes the electron acceptor at the end of this pathway. This generates the hydrogen ion concentration gradient that then goes through ATP synthase to generate out ATP. In non-cyclic photophosphorylation, we utilize photosystem 2. Water gives up electrons to chlorophyll in photosystem 2 thylakoids. This, along with the light, will excite the electrons to flow down the pathway and actually go through photosystem 1 again and go to NADPH, which will go eventually to something we call the Calvin-Benson cycle. This also will cause a hydrogen ion gradient again, and this will then generate out ATP. The light-independent reactions do not require light directly. They are going to use the ATP and NADPH generated by the light-dependent reactions. The key reaction in the light-independent reactions is carbon fixation by the Calvin-Benson cycle. This is why plants can still live in the dark for a period of time. Here's the Calvin-Benson cycle. Again, don't memorize the pathway, but what we see is ATP goes in, NADPH goes in, and what we generate out is glyceraldehyde 3-phosphate. This then can reverse to form glucose 6-phosphate, which will go to glucose. The glucose then can be stored or go directly into glycolysis. Here's a comparison of the three types of phosphorylation: through substrate level phosphorylation, oxidative phosphorylation, and photophosphorylation. So, in summary, energy sources, whether it be light or some sort of chemical energy, will create ATP and electron carriers, which will go through electron transport chains to final electron acceptors. Oxygen if you're aerobic respiration, anaerobic respiration to something else besides oxygen, and in fermentation, organic substances. We can use the energy source to classify organisms from a nutritional standpoint. All organisms then are either chemotrophs that use chemicals for their energy source, or phototrophs. Which one are we? The chemotrophs then will be divided by what the carbon source is. If you use organic compounds such as carbohydrates, lipids, proteins, you're a chemoheterotroph, and your final electron acceptor at the end of the electron transport chain is either oxygen, not oxygen, or an organic compound. If, however, you're a chemotroph that uses CO2 as a carbon source, you're a chemoautotroph. This includes the hydrogen, sulfur, ion, nitrogen, and carbon monoxide oxidizing bacteria. We don't worry about these guys too much because they don't cause disease. If you're a phototroph that uses light as an energy source, and your carbon source is some sort of organic compound, you're a photoheterotroph. This would include the green and purple non-sulfur bacteria. However, if you're a phototroph that uses CO2 as your carbon source, you're a photoautotroph. Then the question is, do you use water to reduce the CO2 to form glucose? If you do and produce oxygen, you are an oxygenic photosynthesizer, such as plants, algae, and the cyanobacteria. If not, you're anoxygenic, which includes the green and purple bacteria. Anabolic pathways are synthesis reactions that require energy and a source of metabolites. Energy derived from ATP is from the catabolic reactions we've just described. Many anabolic pathways are the reverse of catabolic pathways. The reactions that can proceed in either direction are known as amphibiolic. For example, gluconeogenesis, the formation of new glucose. Here we can take pyruvic acid, go back through a pathway to regenerate out G3P, go back to fructose 6-phosphate, which can be converted to peptidoglycan for the cell wall in bacteria, glucose 6-phosphate, which can form glycogen as a storage form of glucose in muscle cells, back to glucose, string those together to form starch, and cellulose. We can also reform fat by taking G3P, taking it back to DHAP and to glycerol, taking acetyl-CoA, reverse beta oxidation, form the fatty acid chains, take 3 fatty acid chains and glycerol, and form a triglyceride. We can also reanimate or transaminate different amino acids in order to reform proteins. So I can take oxaloacetic acid, add ammonia, NH3, and form aspartic acid through amination. Transamination, I'm simply going to take one amino acid, take the amine group off it, put it onto oxaloacetic acid, and form aspartic acid plus alpha-ketogluteric acid. The biosynthesis of nucleotides within cells, the pentose phosphate pathway is going to play a large role here in the production of ribose 5-phosphate that then will allow us to form the pyrimidine and purine nucleotides that make up DNA and RNA. How do we integrate and regulate metabolic functions? Cells can synthesize or degrade, channel, and transport proteins as needed. Cells often synthesize enzymes needed to catabolize a substrate only when the substrate is available. We don't need the enzyme. Why make it? If two energy sources that we can use are available, cells catabolize the more energy efficient of the two first. If you have both carbohydrates and lipids in a cell, you'll use the carbohydrates first because they give you the biggest amount of ATP. Cells synthesize metabolites when they are needed, cease the synthesis if the metabolite is available. Many times this is through non-competitive inhibition of enzyme pathways through negative feedback. Eukaryotic cells isolate enzymes of different metabolic pathways within membrane-bound organelles so that only certain things can happen in certain places within the cells. Cells can use the allosteric sites on enzymes to control the activity of the enzymes through non-competitive inhibition. Feedback inhibition slows or stops anabolic pathways when the product is in abundance. If I already have plenty of tryptophan, I don't need to continue to make it. And cells regulate amphibiolic pathways by requiring either different enzymes or different coenzymes for each of the pathways. Two types of regulatory mechanisms occur within cells, and really this boils down to a single one: control of gene expression. Remember, enzymes are structurally proteins, so we can control the amount and timing of protein production through the expression of the genes. We'll talk about some of this later on. However, once we make the proteins, we then can control the activities through different types of inhibition or through the use of cofactors and coenzymes, and therefore control metabolic expression of those enzymes. Here are all the pathways put together. The yellow arrows are catabolism and the blue arrows are anabolism. You'll notice, however, that cycles tend not to be amphibiolic. So the citric acid cycle and electron transport, you can't bounce the ball back up the stairs and you can't turn the cycle in the opposite direction. Same thing for photosynthesis, it only goes in one direction. But otherwise, most of the reactions we've talked about are reversible reactions. I put this slide in about Earth's energy budget simply to remind myself to mention this. Energy can neither be created or destroyed. It can only change forms. This actually comes from the first law of thermodynamics in physics. This simply shows that all of the incoming solar energy, if you added all these numbers back up again, would equal 100%. So what comes in has to go back out or be absorbed in some fashion. So we can take energy from our food or from the nutrients within the cells and create energy, ATP, to do the work of the cell. Thank you.